So when Bruce Walker founded the Ragon Institute, he wanted to make it a collaboration between Mass General, Harvard, and MIT. But you can tell how where his priorities were, because we're nowhere near Harvard or Mass General. And the reason, I think, we'll see is when we hear from our next speaker, Darrell Irvine, because MIT is a place that's full of innovation, that's been less engaged, I think, in infectious diseases. Now we're starting to see some really, really fascinating things coming from great people there like Darrell.
Thank you, Eric. So thanks to the organizers for giving me the chance to talk here. My lab hasn't been working on TB, but we have been developing vaccine technologies that might have application to TB, and I'd be very interested for feedback from this group. So what my lab is really interested in particular, is working on subunit vaccines and what happens when a vaccine leaves the needle. So we're interested in questions of where do vaccines go, what cells do they interact with, and controlling how they interact with the immune system. And I'm going to tell you two stories in the time I have today of two technologies we've been working on that are trying in part to address the issue that we'd like to make vaccines as subunit vaccines where we have very well-defined safety from purified proteins, but build back into them some of the immunogenicity that we have with whole microbes. So the first story I want to tell you about is thinking about sort of one of the canonical ways to try and do this is to recognize that the immune system has evolved to recognize the particulate nature of microbes, and that actually triggers a variety of features of the immune response, like cross-presentation of antigen. And so for many years, people have thought about formulating antigens as particulates for vaccination, and liposomes are probably the most obvious and most studied strategy to do this. And there are a few examples of virosomes that are essentially liposomal products that have reached licensure in Europe. But we were interested in the question of why liposomal vaccines aren't more immunogenic, at least in animal models. And one of the ideas that we got interested in was that if you look at the compositions of lipids that we make liposomal vaccines out of, they would be expected to be quite unstable in physiologic conditions when they're exposed to albumin and lipoproteins in serum. And so we were looking for what would happen if you designed a lipid-based vaccine that was just more stable and remained a particle when the immune system encountered it because of these advantages that particulate antigen delivery has. And so a few years ago, we came up with this strategy to try and design essentially toughened lipid vesicles by forming first simple liposomes that just have two characteristics. One, that they have a net negative charge on their surface, and then secondly, they have these thiol reactive maleimide groups. So you form a vesicle in the presence of an antigen, you add a divalent cation that makes these negatively charged vesicles fuse with one another, and then you get a multilamellar vesicle. And then while these cations are holding the bilayers together, you add a membrane-permeable dithiol that diffuses between the bilayers and chemically staples one bilayer to the next so that you wind up with these little structures that look like nanoscale rubber tires where you've got 5 or 6 bilayers chemically linked to one another, forming the wall of this capsule with an aqueous interior. And we can either link antigens to the membranes or have it entrapped in the aqueous core, or both. And then you can easily imagine additionally incorporating molecular adjuvants like LPS derivatives like MPLA that could either go in the capsule walls or be entrapped in the interior. And what happens when you form this kind of stable package is you can really significantly influence some of the early steps of antigen processing and delivery in vivo. So for example, if we inject a fluorescently labeled protein antigen in mice subcutaneously and then look in the draining lymph nodes over time, what you'll find is that antigen will flush through the lymph nodes within a few hours. So the exposure window the immune system has to capture an antigen is actually quite short. But in contrast, if you package into these 200 nanometer capsules, they're a little slower to begin accumulating, but what happens is they accumulate over time and build up in the lymph node. They clear out of the injection site in about 7 days, but they form these intranodal depots of antigen that both very substantially increase the amount of antigen that each antigen-presenting cell is able to acquire, and the amount of antigen that each cell has is going up substantially. So the net integral of antigen you're getting into the system is much greater. And because it's a particle, we additionally have this feature that compared to a soluble vaccine, even with an adjuvant like monophospholipid A that's not known to particularly promote cross-presentation of antigen, if we immunize mice with soluble ova as a model antigen, whole protein mixed with MPL, you don't elicit much of a T cell response in the blood. But if we immunize with these capsules, we can see an expansion on each immunization here, reaching up to about a quarter of the CD8 cells in the blood being specific for a single epitope. So really the kind of response that previously people would only see with a viral vector. And these cells are functional, so they kill target cells, they're producing cytokines at high levels, and we're getting CD4 responses in parallel. For humoral immunity, there are a couple of very interesting things that happen. So these vaccines, and we think this is linked to this depoting effect in the lymph node, you drive much stronger differentiation of follicular helper T cells. And so, for example, using a malaria antigen where with a variety of traditional adjuvants, you get this characteristic early peak response that dies out, we could immunize with the same antigen in these capsules at the same dose and get this very dramatically long-lived immunity lasting more than a year in these mice. And further, you could drop the antigen dose by tenfold, which would ablate the response to all of these other adjuvants, but still keep the response with the capsules. And that dose-sparing ability then lets you do things like draw out high avidity B cells so that if you measure the binding avidity of these antibodies that are being generated, they tend to show higher avidity to the target. So both humoral immunity and cellular responses, at least in small animals, are significantly promoted by this approach. More recently, we got interested, and this might be of interest in the context of TB, of whether the stabilized nature of the capsule would let you also pursue non-invasive routes for vaccination. So we looked at whether these capsules would survive administration intratracheally to mimic a pulmonary vaccine with the goal of targeting these very high levels of dendritic cells and macrophages that you have lining the airways, which can all transport antigen to the lung-draining lymph nodes. And here what we saw is that the capsules do in fact trigger rapid acquisition by macrophages and dendritic cells in the lung tissue so that 24 hours after administration, while the soluble antigen has been cleared away, if we're delivering it in these packages of lipid capsules, it's still around at 24 hours and takes a few days to clear away. And then if we look 4 days after immunization, while we can barely detect antigen in the draining lymph nodes in the soluble vaccine, you see this massive accumulation of antigen that when you go in at finer detail are packages of particles in antigen-presenting cells that have transited to the draining lymph node. And that then in turn impacts significantly the kind of cellular immunity you elicit with this vaccine. So if we give a pulmonary vaccination with ova in mice in the lungs, where we're delivering as coadjuvants poly-IC and MPLA as two TLR agonists, boost at day 28, and then follow first by peptide MHC tetramer staining to follow antigen-specific T cells in the, this is looking in the blood. You see that the capsule vaccine expands the cells more and they can be boosted. And after a single boost, we get this stable memory population that's present both in the blood and the lungs. And strikingly, and we believe this is linked to the high levels of antigen that we're getting to the lymph node, these are highly biased toward an effector memory phenotype, where we have a pronounced population both in the lungs, in the blood, the spleen, and as I'll show you in a minute, in distal mucosal tissues of highly effector memory biased T cell population. Now, we had originally been focused on applying this hopefully to HIV, and one of the interesting things about this pulmonary vaccination potentially for HIV vaccine development is the fact that if we do trafficking experiments where we transfer in a small population of ova-specific T cells that are expressing luciferase into these recipient animals, and then track the antigen-specific T cells over time following immunization, if we give pulmonary immunization with the nanocapsules or the same vaccine in soluble form versus sub-Q vaccination. In the sub-Q vaccine, all the action's happening in the draining lymph nodes. We gave tail-based immunization here, so the inguinal lymph nodes are lighting up a few days out. In the pulmonary vaccine, we have the thoracic lymph nodes lighting up. But a few days later, once the prime T cells begin leaving the draining lymph nodes, there's a really dramatic impact of administering the vaccine in the airways, where following expansion, these cells disseminate to the gut, to the reproductive tract. There is a large population that are in the lungs. If we take the intestines out, we see all the Peyer's patches lighting up, and especially the cecum in these animals that received the nanocapsule vaccine. There seems to be qualitatively a similar trend, but much weaker with the soluble vaccine given in the lungs. And the sub-Q vaccines do a very poor job of eliciting this kind of disseminated immunity. So there's, at least in the small animals, an ability of the lung vaccination to promote this distal mucosal response. So then in terms of the degree of protection this kind of vaccine elicits, we did protection studies where we'd immunize in the lungs with an HIV gag peptide boost on day 28, and then come in with a challenge with vaccinia virus expressing, and I'm sorry, I should say this was SIV gag, coming in with an SIV gag expressing vaccinia virus. And if you look at the pattern of viral dissemination in these animals 7 days later, we see that the nanocapsule vaccine lowers the viral burden in the lungs by 2 orders of magnitude, and it completely blocks dissemination systemically to the ovaries, where the soluble vaccine in half the animals has completely failed to block systemic vaccination, systemic dissemination. And that in turn translates into a very different outcome in terms of survival. So the same dose of antigen and adjuvant given in soluble form in the lungs is completely unprotective, but when administered in this capsule form, protects 100% of the animals. So we think there's some promise here based on the change in the early dynamics of vaccine delivery here. So by making a stable particle, we enhance the uptake by antigen-presenting cells in the initial tissues we deposit. That gives us better trafficking to the draining lymph node. It seems that one of the features of these capsules is they create this intranodal depot of antigen, which, interestingly, if you dig into the old literature of how alum works, alum as a depot is not important to persist in the initial tissue where you deposit it, but there's some evidence that alum makes depots in lymph nodes of antigen. So we may be sort of recapitulating some aspects of that well-known adjuvant. And this leads to several changes in the immunology. So we enhance TFH differentiation and germinal center induction. That I didn't have time to show you. You promote cross-priming of CD8 cells, and at least in the airways, you can promote this very strong effector memory T-cell bias response, which in the vaccinia case we showed was responsible for protection in the lungs. Okay, so that's one strategy that we've been looking at for a few years. And with the rest of my time, I want to describe a more recent strategy that has been focused on the problem of peptide-based vaccines. So for peptide subunit vaccines, a real problem is this issue of anatomical distribution, where you inject your vaccine in the muscle or the subcutaneous tissue, but all the action has to happen in the draining lymph node. And we not only have this issue of poor antigen capture in lymph nodes, which I already alluded to, but when we're giving a subunit vaccine that requires some adjuvant to promote the immune response, you have the problem that if we use a molecular adjuvant, like in this story I'm going to focus on CpG DNA, these single-stranded DNA oligos that trigger toll-like receptor 9, whatever material isn't captured in the draining lymph node eventually ends up in the systemic circulation, and that leads to the kind of inflammatory toxicities that are completely unacceptable for a prophylactic vaccine. So we're interested in, is there a way to design this kind of molecular vaccine such that it gets to the lymph node, it gets there very well, and it doesn't go any farther? And a postdoc in the lab got interested in the idea of stepping back for a second and asking, are there any existing clinical procedures that already are known to deliver compounds to lymph nodes in humans? And he focused in on the process of sentinel lymph node mapping that's used in oncology. So in breast cancer and melanoma patients, one of the common procedures that's done is at the resection site where a tumor has been taken out, the surgeon will inject a dye that's taken up very efficiently into the draining lymphatics and then is captured in the draining lymph nodes and optically stains them so the surgeon can resect the lymph node and stage the tumor. And though those dyes that are small molecule compounds were all originally identified empirically, it's now understood that one of the design rules to make a great sentinel lymph node mapping dye is to make a compound that binds very tightly to endogenous albumin. And the thinking is that you inject these dyes, they bind to albumin, that blocks their dissemination into the bloodstream and sends them one way into the lymph nodes. And now there's some evidence that adducted albumins become a target to be scavenged in the lymph node, and the cells that are doing that scavenging are, as you might expect, antigen-presenting cells, so the exact cells we might want to send a vaccine to. So Haipeng Liu, a postdoc in the lab, started working on the idea of asking, could we mimic what happens with sentinel lymph node mapping dyes to send a vaccine to lymph nodes more efficiently? So he focused in on the idea that albumin, one of albumin's main functions in the body is to serve as a fatty acid transporter. It has 7 different fatty acid binding pockets. So he started designing molecules where he'd take CpG DNA or a peptide antigen and link these to a lipophilic tail optimized for efficient albumin binding, with the idea that you'd inject these amphiphilic molecules, they would associate with albumin and undergo the same sort of lymph node targeting process. So we made a series of amphiphiles, and I'll show you first a little bit of data with CpG as a model molecule, where we put a fluorescent tag on CpG. We put different kinds of lipid tails on the back end, cholesterol, a single acyl tail, a diacyl tail, and then we looked for in vitro biochemistry evidence of association with albumin. And so one of the assays you can use is a size exclusion column where we run serum through and we get this major peak, which is albumin. If we run free CpG through, it elutes at a different location. If we mix CpG with serum, CpG still elutes in the same place. But if we, for example, take this diacyl-tailed molecule, on its own in this column, it forms micelles in water, so it makes an elution peak to the left. But if we mix it with serum, which is the green here, about half of the material is eluting as a micelle, and half of the material co-elutes with albumin. And in FRET studies, we showed that that's actually a molecular association of this molecule with individual albumin compounds. And by a series of other physical chemistry assays that I'll skip for time, we showed that if you have a long saturated diacyl tail, a very lipophilic tail, you can drive binding to albumin. Now, in vivo, there's a sort of 3-arm equilibrium we're worried about. So if we take these amphiphiles and put them in water, they want to make a micelle. If you expose them to serum, serum will break up the micelle and bind to the tail. But if you have no serum around, these amphiphiles are also happy to jump into cell membranes. And this we didn't want to have happen because we worried that this would block transit to the lymph nodes if it's hanging up in the tissue. So we also realized that you have to make these molecules with a long enough polar block so that they're carried into solution and biased away from cell membrane insertion. So sort of the design rules are make a long lipid tail, make a long enough polar block to keep the molecule in solution, and this we proposed would drive to this state when we inject in vivo and promote lymph node trafficking. And just as an aside, so for CpG, it's already a pretty polar long, it's a 20 base polynucleotide, so it's already long enough to avoid cell membrane insertion. For peptide antigens, which themselves could be quite hydrophobic, we introduce a 2 kilodalton polyethylene glycol block here, and that serves the purpose of keeping this thing in solution. Okay, so we made a series of molecules that better or worse interact with albumin. So then we tested whether that influences trafficking in vivo. And one of the assays we'd use is we make fluorescently tagged versions of these amphiphiles. You inject them sub-Q. You take out the draining lymph nodes at different time points after immunization. And just first you can look at the whole lymph nodes just excised from the tissue by fluorescence imaging. And one of the first things I was really shocked at is how poorly CpG, which is a decent adjuvant, is actually accumulated in lymph nodes. This is 24 hours after injection. and there's very little detectable. By histology, we couldn't even find fluorescent CpG in many lymph nodes. If we make CpG linked to a cholesterol tail or a single acyl tail, it really doesn't change the picture much. But if we take that diacyl tail that binds to albumin very well, we can increase the accumulation in lymph nodes about tenfold, and it's more than we get if we use IFA or liposomes as carriers. And in histology, you see the same sort of thing. So this really changes dramatically the ability of this compound to get to lymph nodes and accumulate there. Now, having identified compounds that get to lymph nodes efficiently, then we asked, what does this do to the immunology of these factors? And as I said at the start, one of the issues with adjuvants is you want to get them to the lymph node, but you don't want them getting into the circulation. And it's known that as CpG as an example, even in sub-Q injection in patients, within a few hours you see systemic inflammatory cytokines. And we see the same thing in mice. So if we give a sub-Q injection of CpG, in 2 hours we can see high levels of IL-12, TNF, IL-6, and so forth in the serum. These amphiphils that are trapping in the local lymph nodes but not getting farther completely block that inflammatory syndrome. And if you repeatedly inject CpG in soluble form, you can get splenomegaly. That's completely blocked with the amphiphile. That might mean that we just killed the activity of the molecules, so we also looked at vaccine responses. And first we did experiments where we just asked what happens if you target the adjuvant to the lymph node and give a soluble protein antigen. So here again we're using OVA as a model antigen. We prime on day 0 with soluble CpG or amphiphile CpG, boost with the same formulations 2 weeks later. At 3 weeks we measure antigen-specific T cells in the blood, and what we see is that here's soluble CpG, and CpG is known to elicit a small amount of cross-presentation in mice. So there's a detectable response. It's about 1% of circulating CD8s that are specific. You can increase that 30-fold with the optimal lymph node targeted molecule, giving the same dose of antigen and adjuvant. This series of molecules are different variations that get to the lymph nodes at different levels depending on their structure. And what was really fascinating is if you plotted lymph node uptake of that same series of adjuvants, the molecules that get to the lymph node the most efficiently are the ones that are giving you the T cell response. There's essentially a perfect correlation where as we varied structure of these adjuvants, the better they got to lymph nodes, the better they primed T cells in an almost linear fashion. So there really is a dramatic impact of how well you get the adjuvant there. And then you could ask if it's good for the adjuvant, is it good for the antigen? So we made amphiphile forms of several different peptide antigens, including epitopes from HIV GAG, from melanoma antigens. This is from HPV. And in each case here we're comparing, we've immunized twice, and then we're looking at cytokine-producing T cells by intracellular cytokine staining. In each case here we have the soluble vaccine and then the same amphiphile form of the same compounds, and you see that in each case you're increasing the response between 5 and 10-fold. And for protective efficacy, we're a little farther along in testing this in the setting of cancer. So I'll show you just as evidence for functionality of the CTLs we're eliciting. If we, in a cervical cancer model or an aggressive melanoma model, where we administer tumors subcutaneously and let them grow, the traditional soluble vaccine will slow the cervical cancer a little bit, but we can actually regress these large tumors with repeated vaccination using the amphiphile. In the melanoma model, where a traditional vaccine has 0 impact, you can actually see some slowing down of tumor growth. So now where we're trying to move with this in HIV is to combine this with some other efforts going on in the Ragon Institute. Arup Chakraborty has been developing a theoretical approach to analyze sequences of HIV virus and predict how fit virus will be as a function of sequence, and thereby define epitopes to target which would maximally impact fitness while avoiding compensating mutation escape from immune pressure. And so we're in the process of launching a non-human primate study to see if this technology will work in large animals, where we're going to overlay Arup's fitness predictions of where to target in GAG and other proteins alongside known hotspots where macaques robustly respond to HIV sequences. And so, and I'll just show you one example here to show that in these long peptides that require cross-presentation, even in mice, we can again see very robust intracellular cytokine responses in animals using amphiphile forms of these long peptide sequences. So what we're excited about with this approach is the simplicity here, because you're talking about molecules that may be possible to simply engage an existing contract manufacturer to add these lipid tails to an existing solid phase synthesis process to make molecules that we would predict would both be more potent and more safe for vaccination, and will hopefully be able to see the same responses in large animals and have something that would be simple and cheap to manufacture. So I'll stop there and just acknowledge again the work in the first story of the lipid capsules was really driven in the lab by a postdoc, James Moon, and the mucosal pulmonary vaccines by grad student Adrian Lee. And the second story I told you about, the amphiphile vaccines, was the work of a postdoc, Haipeng Liu. And we're grateful for support from the Ragon Institute and the NIH on these projects. Thanks for your attention.
Yeah, very interesting stuff. And I'm wondering, the systemic effect that you saw with pulmonary administration, are those particles stable enough to get through the gastrointestinal tract?
So we looked for that because we had seen some reports that proteins given in the pulmonary system would get to systemic sites. We could detect neither the capsules directly nor evidence of antigen presentation at anywhere except the lung draining lymph nodes. So we don't think they get beyond the lungs.
Go ahead, Sarah.
Oh, can I just ask a related question? If you deliberately deliver them to the gut orally, will you also get robust tissue-specific?
We've only done a couple of experiments there, and I'm not sure these particles are stable in the gut. That would be my short answer. I think we've got a lot more to explore there, because there are a lot of compositional games we can play with the capsules. But early on, we compared oral and pulmonary vaccination, and the pulmonary was much stronger, so we sort of focused there.
So I'm maybe totally confused, but one of the things that has always really disappointed me is that even with the best new compounds, new small molecules for treatment of TB, while you get great activity in the lung, whether they be given by aerosol or orally, the most difficult place it seems to eradicate the bugs are in the spleen. So I'm just wondering, with this approach, which is fantastic, by the way, really impressive work, but what about, I guess, the residual infection in spleen, and maybe if you accept the work that the Stanford group's done showing persistence in mesenchymal stem cells, which I know there may be some that maybe are not ready yet to accept that. But what do you think about that in terms of this approach for vaccines?
Well, for systemic targets, it's actually, I think, for the systems we're looking at here, we have an easier time promoting systemic, say, splenic protection. With both sub-Q and pulmonary vaccines, we're getting quite strong memory populations established in spleen. Yeah.
Sorry, I missed that.
Yeah.
If I can, Sarah asked my question, but if I can expand on it a little bit, if one of the concerns with delivering pulmonary vaccines is induction of reactive airway disease, are there other mucosal sites you might go to and get the same sort of cross protection?
Go to the lung from it. Yeah, we've started thinking about that. I've heard tell of some data that in humans, maybe even skin vaccination is promoting lung dissemination. We haven't really explored it yet much, but I think that's definitely an area to be looked at further.
Nasal vaccination with the lung.
Yeah.
So it was very exciting. I think that few people appreciate the importance of formulation, you mentioned CPG. The dose in humans in the current Hep B vaccine is 3 milligrams. So this dissemination of a hydrophilic small molecule to cause toxicity can kill even though $300 million is spent developing this, it can kill an adjuvant. Having your formulations go in the cell is very important. But can you make an SUV or a multilamellar vesicle a little bit smaller so that you can have endpoint filtration as you're in your process? Because 200 nanometers pushes the...
Yeah, that's a great question, and also equally important for this to be viable. Actually, I mentioned 200 nanometers because the majority of the work we've done in the lab has been with those larger particles, but recently we have gotten, so this cryo is actually, yeah, so we recently figured out how to get them smaller, and now these are sterile filterable, yeah.
Okay, appreciate that. Thank you very much.